So welcome to demo video 4 in our course B0, B10, Python for AI, where we're talking about protein language models in an applied sense here. So we're thinking about how protein language models can take the abstracted sequence of amino acids and from that to create tokens from them to weight them differently to identify ones that have a bigger impact on the structural patterns of that particular protein, and then model these in more effective manners, such as looking not just at the secondary structure, but the tertiary and quaternary structure in terms of how proteins or other biological molecules interact. Key to this is using transformers, which allow transformers to identify which sequences are more important. And they can identify, say, for example, that certain sequences are related to each other, even if they're far apart. And the attention scores are based on the Q, K, and V parameters, which are the query key and values parameter, which are a transformation to identify what am I looking for, what information what do I offer, and what information do I carry. ESM2 is a protein language model, which is trained on a large number of sequences. It hasn't seen structure or function labels, but is produced by predicting masked residues. So it's basically taking an existing protein sequences, putting ends for removing the protein amino acids for certain regions, and then trying to see if it can infer what ones are missing. So here we're going to use for our ESM. If we install that, it should be smooth and quick. We also need to install ESM as well and PyTorch. And what we're using is a pre-trained one, which is listed here, which is fairly lightweight and suitable for a CPU environment. And that's running away there. It's downloading that information and saying that ESM is loaded. So what we're looking at is three amino acid sequences for three beta-lactamase, TEM1, OXA1, and BIM2. BIM2 is a metallo-beta-lactamase, and if you look at the sequences here and you align them, you'll see that they're quite divergent from each other, although they have obviously shared motifs like this FAA segment here in the TEM1 and OXA1. Other regions of the proteins, they're quite dissimilar. And what we can do is take these sequences, and what we can do is send these sequences to our model to ASM to then apply them to this, where we're treating them as the variable I here to extract their embeddings. So now they're extracted, and now we can use the package sklearn to look at the embeddings which are mentioned there to infer their similarity based on plots to look at how similar they are. And what we can see is that TEM1 and OXA1 show some differences, and BIM2 and OXA1 are more similar to each other. That's kind of what we see here in terms of their structural similarity. And what you see is they're quite high matches for each other, despite the divergent amino acid sequences. We can also apply AlphaFold as well. And what this does, it takes the protein, the amino acid sequences of the molecules of interest. It uses a fair, fair pair former, a transformer on amino acid pairs, to look at their spatial relationship, and does this for all possible pairs in the sequence. And then uses a diffusion model to estimate the variation that we might have in these different predictions, to see which ones might be more real or less real, and does this by training in terms of going forward prediction from the real structure to add noise iteratively, step by step to get pure noise, and also reverse by taking pure noise and denoising iteratively to get the predicted structure eventually. So what I was trying to learn is the denoising direction. And it starts from random noise and iteratively tries to get the correct prediction. So AlphaFold 2 just gave a point estimate, whereas with AlphaFold 3 we can get a distribution of outputs, letting us estimate how confident we are of the prediction. So what we're doing here is taking our resistance data for zone of inhibition, and we're looking at the level of accuracy present what we see there is that you have the original data is distributed here. If we had light noise, we can see the separation between the two groups, and as we add more noise, we can see that they become more mixed. So what we can do is go to the AlphaFold server, sign in with our account, and then paste in one of the sequences that are present, and that will create a map that I will show you now. So these are examples where we have the TEM1 is the first amino acid sequence, OXA1, and then BIM2. And if we click on the first one, which is for TEM1, what we can see eventually is the 3D structures here. And what is worth noting is the relative orientation, if I get this in the right orientation. You can look at the structure similarity between the different proteins that we have here. So you can see if we look at the confidence scores across the regions, is that there's the two arms here are low predictive accuracy, whereas the internal structure, you can see those beta sheets. Some are confident, some are less confident. You can also focus in on predictable residues. For example, this green area here indicates high confidence regions, which is the ones highlighted in pink here. And if we unhighlight that, you can see those are the ones in blue. If we look at the first part here, you see that has generally low confidence. And we can look at bits here that have maybe better confidence in part as well. And then if we look at our second prediction, so this is for OXA1. What we can see eventually is that it has high structural similarity with, that looks incomplete. High structural similarity. Let's go to something we had to miss there, to VIM2. This is more like it. So you can see again those two orange arms where this pure predictive power, and then again at sites, at the first few sites at sites maybe 20 to 92, we get a high level of accuracy. So you can see those areas in pink there that are highlighted. There are ones with high level of accuracy. They're the ones in blue here. Again, you have this second region of high accuracy here, again in the core, and then you have more at the end. If you go back then to the original one that we had before, you'll see that the structures are quite similar to each other. We can sort of see this down here. If we look at the images of each of them, this is TEM1. So you can see there's conserved regions here on our left that are repeated when we look at the other ones. So we got one here, a moderate one here, and a smaller one here. And if we move to OXA1, again, you see the structures are very, very similar to each other in terms of they're very similar to each other, and the aligned residue scores again reflect that too. If we look at VIM2 next, you can see that again that there's a high level of structural conservation, even though the amino acid sequences actually diverge quite a bit, and those regions are present throughout the protein. So that kind of ends our the practical side here. So what we saw is that the EDA shows that the the differences between the groups was poor. We saw that we were able to distinguish the groups using support vector machines, gradient boosting, and neural networks and deep learning. We saw that the differences in terms of our data set here weren't too different from each other. So even if we have very advanced models, we can't necessarily make magically perfect results from them. It's simply not possible with the data set that we have. And we're able to use the AUC metric to estimate more accurately how good they are. What we saw there at the end was that we can create embeddings in ESM2 to model how structurally similar different proteins are, and then how you can also visualize them effectively with AlphaFold 3. So that completes the demo video. Please see the introductory videos if you've missed out on the theory for this portion.